So I know it's hard, but don't mentally check out for the next 2 days. And everybody's already thinking about spring break. Anybody have any questions? Okay, it's been a while since we've had a reading quiz, and so there will be a reading quiz over a very short section of the last part of the chapter, 167 through 171. We may or may not be able to kind of introduce that topic today, but I want to make sure that we have the chance to have you look over that and read that information. So Monday we will do that. We will have the, I'll hand back the lab quiz and go to the lab quiz, and then we will do a little bit of introduction to the PGLO lab, okay? Remember that we're going to switch 2 labs. You don't have to do your lab presentation the week you get back. We'll do the PGLO lab, and then you'll have that week to work with your partner, and those presentations will be the following week. So I don't know if you want to say anything the Monday after break about the presentations, but you already have, so.
Yeah, I don't think so.
Okay. Okay, let's do a little review before we head off into the last material regarding photosynthesis. What is this whole group of molecules consisting of all of these chlorophylls and this reactive center? What do we call that? It's a photosystem, okay? And this particular photosystem is designated photosystem number 2. And what we'll take a look at first is the events that happen in photosystem 2, and then see how that connects with the events that happen in photosystem 1. Both photosystem 2 and photosystem 1 are part of these light-dependent reactions of photosynthesis. So these are taking place within those thallicoid membranes. And when these photosystems are completed, we really need to account for what happens to water, how oxygen is produced, how ATP is produced, and how reduced NAD is produced. Okay, so we just dig in a little bit more detail into how that happens. And we'll spend a little bit more time talking about the process of making ATP particularly. Okay? So we've got all these clusters of molecules. Typically there are 200 to 300 chlorophyll molecules that are packed around that reactive center. Okay? What you might think about this as, in terms of analogy, is let's say we've got a cluster of pool balls sitting in the middle of a table. And then additionally, spread around on that table are all of these different little other balls. Those would represent these antenna pigments. And then this would represent the reactive center. So if I take a cue ball and I hit it with my cue and I strike one of these balls, then we can start getting collisions occurring between these particular balls. And the momentum from this ball strikes this ball, strikes this ball, strikes this ball. But eventually, one of those is going to collide with the reactive center, and that's going to cause this electron to be elevated to a higher energy level. So these aren't losing electrons, okay? When their electrons are excited, they stimulate another chlorophyll, and then those electrons drop back down to ground state again. Okay? But here now, an electron is elevated, and then grabbed by this primary acceptor. Okay? So all we've done really is we've converted the energy of a photon of light into an excited electron in the reactive center. Now, this isn't stable, is it? And one of the tendencies for this electron to do would be to become stable and fall back down to ground state again. What do we call that process? An elevated electron just drops back down to its original stable state. And we emit light as a form of energy. Remember from Tuesday? Fluorescence, good. That's called fluorescence. But that doesn't get us anything in terms of productive energy for a plant to grow. Fluorescence doesn't help them grow. So, once that primary acceptor, okay, let's continue for a second. That primary acceptor is going to then donate that electron to an electron transport chain, and that will be contributing to the development of a hydrogen ion gradient across the membrane, across the thylakoid membrane, and that can be used then to make ATP. What do we call the process of making ATP using electron transport in photosynthesis? It's called photophosphorylation. Remember, oxidative phosphorylation is the process that we use in cellular respiration. Okay, so if we keep doing this, the photons of light keep bombarding this photosystem too. And we keep punching electrons up here and transferring them here. We're going to run out of electrons over here pretty soon, right? This compound will become oxidized. Its electrons will become depleted because they're being stolen away and donated to the electron transport chain. So we need to replenish those electrons, and that happens by water split. So in the presence of light, water is split, oxygen is released, some protons are released. Keep that in mind, that's going to be important later. And then the electrons are used to replace these electrons that were lost from the reactive center. So do you see the essential importance that water has in terms of keeping this particular process going right here? Okay? So, we've accounted for so far the role that water plays, where oxygen comes from, and where ATP comes from. While this is happening, at the same time, photosystem number 2 is also involved in photosynthesis. Antenna pigment molecules, the chlorophylls, are excited by light. Light energy basically is transferred to this reactive center, and those electrons are boosted up to a higher state. So we're losing electrons from here, but we're replenishing them from here. When they're grabbed by the primary electron acceptor, they're then donated to NADP. And to reduce NADP, we need protons and electrons before we really produce the reduced NADP. So we're going to be using some protons up here. We're going to be producing some protons here. And we're also going to be making a hydrogen ion gradient here during this electron transport process. So basically, light allows electrons to be stripped from water and move through the two photosystems. Ultimately, those electrons end up as part of this reduced NADP. While these photosystems are cooperating, we also produce ATP by photophosphorylation. So those are the light-dependent reactions. And I can pretty much guarantee that there will be several questions on your next exam that will ask you to be comfortable with events that happen in photosystem 2, events that happen in photosystem 1, how those two are connected, and how those two photosystems explain the various products that are produced during the light-dependent reactions. So here's situations where if you could reproduce this diagram, if you could sit at a board and kind of draw out this diagram and explain it to somebody else, you would understand the light-dependent processes of photosynthesis. Okay. Hydrogen ion gradient. So during those light-dependent reactions, we have a buildup of a hydrogen ion gradient across the membrane. This is the thylakoid membrane. Remember that the membrane wraps around a space. So inside of that membrane is a space. So this is the thylakoid space. And this is the stroma out here. Okay? Stroma, thylakoid space. A compartment, a membrane, and a compartment. So we build up a hydrogen ion gradient. High H plus builds up inside the thylakoid space. So we start accumulating H pluses here. And we get lower H plus in the stroma. What are those protons going to want to do? They're going to want to move from their high concentration to their low concentration. But thylakoid membranes are impermeable to those protons, unless there is an ATPase channel protein. So they flow through the channel protein. A little bit different wrinkle here. For every 3 protons that flow through this channel protein, we make an ATP molecule. Now, in photosynthesis, there are actually 3 events that contribute to the formation of this hydrogen ion gradient, building up a lot of protons here and less here. In cellular respiration, there was only one event, really, and that was the electron transport system. In photosynthesis, the electron transport system also contributes to the hydrogen ion gradient, in exactly the same way as it did in respiration. There are 2 types of carrier molecules in the electron transport chain. What are the 2 types of molecules? Hydrogen acceptors, donors, and electron acceptor donors. Okay. And they are asymmetrically distributed in the membrane. So the net result is that we pump hydrogen ions from here into here as these electrons are flowing through that electron transport chain. Exactly the same way we did in cellular respiration. Okay, that's event number 1. Event number 2. We're going to be reducing NADP in the stroma. Okay, it's out here. To reduce NADP, we need some protons and we need some electrons. Where are those electrons coming from? They're coming over here from this photosystem number 1. Okay, photosystem number 1. So it's donating the electrons to NADP. And along with some protons that are picked up from the stroma, NADP becomes reduced. Why is it important that the NADP is reduced in the stroma? Because this is going to be used in the Calvin cycle. And where does the Calvin cycle occur? In the stroma. Okay? So we're making this thing that the Calvin cycle needs in the same place where the cycle occurs. So this is going to use up protons here. So that will decrease the concentration of protons in this compartment. And then the final event is the splitting of water, and that happens in the thylakoid space. So in this space right here, water is split. Oxygen is produced. Electrons are released. Remember where they go? Photosystem 2. And protons are liberated. So that's going to add the protons here. So the electron transport chain pumps protons from here to here, lowering the concentration here, increasing it here. This process depletes protons here, so it decreases them here. And this process increases protons in the thylakoid space. And we've got our hydrogen ion gradient that can be used to make ATP. So, this just kind of shows you systematically what's happening and kind of shows you a little bit nicer, more detailed point of view of what's happening in these light-dependent reactions of photosynthesis. We've got our thylakoid membrane, we've got our photosystem number 2, we've got our photosystem number 1, our electron transport, buildup of hydrogen ions here as a result of electron transport and splitting of water, the depletion of hydrogen ions out here from the reduction of NADP, and then using that built-up hydrogen ion gradient to make, ATP. So the high hydrogen ions in here, coming back out here again, make our ATP. And both of these products then participate in the Calvin cycle. The Calvin cycle is located out here in the stroma of the chloroplast. So it's really a pretty nifty system if you think about it. Because ultimately what we are doing is we are converting the energy of light into the energy of chemical bonds. And then ultimately we're going to use that chemical bond energy to convert or fix inorganic carbon dioxide from the atmosphere into organic glucose or other carbohydrates, which are considered to be biomass. They're part of the substance of living organisms, of plants. So this compares and contrasts kind of mitochondrial structure and chloroplast structure. They both use the same basic process to make ATP, but remember that things are just kind of reversed. Okay, the hydrogen ion gradient builds up on opposite sides of the membrane in photosynthesis and respiration. So don't confuse that, okay? What is this sequence part of? What process? This is the first few steps of glycolysis, okay? Glucose converted into glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-biphosphate, and then either of these can be produced, but this is converted into this. So basically we make a lot of this glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is abbreviated on the next slide. We're going to see G3P. So keep that in mind. If we can make this by another process, theoretically, do you think it's possible that the cell might be able to then convert this into this, this into this, this into this, and make glucose out of this? Yeah. Yep. A lot of times chemical reactions are reversible. Well, as it turns out, this is the primary product that's produced by the Calvin cycle. Calvin cycle cranks out a whole bunch of this G3P, which is essentially a half of a glucose. Okay, 3 carbons here. And so if you stick 2 of these together, you make some glucose. Okay, so this shows you the relationship between the light-dependent reaction of photosynthesis and the light-independent reaction of photosynthesis for the Calvin cycle. Okay, so we've already gone through this in quite a bit of detail, and we produced this during the Calvin cycle. The Calvin cycle is just a bunch of biochemical reactions. Every step in this process is regulated and controlled by an individual enzyme. And there's one particular enzyme that we're going to talk about a little bit more in detail in just a minute. But basically, what the Calvin cycle does is allows the cell to convert inorganic carbon dioxide, which has been diffusing into the plant leaves, if the stomata are open. There's a certain amount of carbon dioxide always in the atmosphere. Okay? We're breathing it into the atmosphere continually. There's not a whole lot. There's only like 0.03% CO2 in the atmosphere, although it is rising. But that's still enough CO2 to really power this incredible process of photosynthesis. CO2 enters the Calvin cycle. And as a result of the Calvin cycle, the carbon dioxide is eventually converted into G3P. Eventually, this can be converted into glucose. This can be converted into starch. This can potentially be converted into amino acids and fatty acids. It can be converted into sucrose, the disaccharide. Sucrose is the main transport sugar that we find in plants. So the leaves produce glucose and then converted into sucrose, and that's what's transported throughout the plants down to the roots, to the stem tissue. But this process of turning inorganic CO2 into organic carbon requires energy and requires hydrogen. As the ATP is used, ADP is produced. That goes back here and works in the light-dependent reactions again. As the NADPH is oxidized, it then can go back and work in the light-dependent reaction as well. So these two systems kind of recycle for one another. And they really are codependent on one another. Now, biochemically, the Calvin cycle is a nightmare. Larry laughs. What we're going to do is we're going to show you kind of an abbreviated, dumbed-down version of the Calvin cycle. And even the amount of detail we're going to show you, there are certain things that I want you to concentrate on and certain details that aren't quite as important. Okay. So here's our Calvin cycle. Okay, remember what the input is, CO2. What's the other input? ATP. What's the other input? Reduced NADP. The output is this G3P, basically half of a glucose molecule. Okay, here are some important steps in the Calvin cycle. What we're going to do is we're going to kind of assume that 3 CO2s enter the Calvin cycle simultaneously. In reality, that's really not what happens. Actually, the CO2s enter the cycle one at a time. But it makes much easier to understand if we make this kind of bad assumption. Okay? The first phase of the Calvin cycle is called carbon fixation. When we talk about carbon fixation, we're talking about converting inorganic carbon to organic carbon. Something has got to grab that CO2 to get this cycle going. The compound that grabs it is this 5-carbon sugar called RUBP. RUBP stands for ribulose bisphosphate. It's got a couple of phosphates. And we need 3 of these to grab 3 CO2s. RUBP plus CO2. And we form this compound that's just called, they just call it a short-lived intermediate. We're not even going to give it a name right now. But when this is grabbed with this, we form this 6-carbon short intermediate compound that barely stays around for any time at all. It's immediately converted into something else. Okay? So really, this is kind of the first stable compound of this being added to this, is this 3-phosphoglycerol. Okay. You might notice that they're giving you numbers of molecules right here. What they're trying to do is account for carbons. So this is a 5-carbon compound, and there are 3 molecules in this, so that's 15 carbons, right? There's 3 more carbons. So this should have 18 carbons, and it does. 3 carbon compound, 6 molecules of that. So we've accounted for all of our carbon. This enzyme is an important enzyme. The enzyme that grabs CO2 and starts the Calvin cycle is this enzyme called RUBP carboxylase, or if you prefer, it's usually referred to by its acronym Rubisco. Okay, so this little box right here represents the enzyme Rubisco. It grabs the CO2 and fixes it to this organic molecule to make this first stable product of the Calvin cycle. Now, don't forget about Rubisco, because in your reading, if we don't get to that today, Rubisco is going to come up again, this enzyme. Because Rubisco can participate not only in a process called carbon fixation, which is essential for photosynthesis to take place, but this same enzyme is also involved in a process called photorespiration. And photorespiration is kind of bad for the Calvin cycle. Okay? The purpose of the Calvin cycle is to make sugar. Photorespiration kind of prevents the Calvin cycle from doing that. Seems kind of odd that one enzyme can do 2 different things, one of which is productive and one of which is destructive. But that's going to be important to wrap your head around in reading those 3 or 4 pages you have for the quiz. Okay, so the rest of the Calvin cycle really is all about getting back to here again, because we always have to regenerate this again, so this can keep going, and siphoning out at some point that half a glucose molecule, that G3P. And along the way, we're going to use ATP, and we're going to use reduced NADP. Let's just kind of see kind of what happens here. Okay? So, this molecule is converted into this one. That requires some ATP. This is converted into the G3P. That's going to require some hydrogen from the reduced NADP. One of these 6 molecules of G3P is siphoned out of the Calvin cycle. So now we've got half a glucose basically. And this could be used to make more glucose or other carbohydrates. The remaining 5 are going to be recycled back into this. To the carbons added. Okay, what do we got? We've got 6 of these. That's 18 carbons. We're pulling 3 out of the cycle. Those are the same 3 that came in. Okay? So now we've got 15 carbons left to regenerate this. Does that add up? Yeah. 3 times 5 is 15. So this is converted into this, and this is finally converted into this, and that requires some more ATP. So look at the energy demands for making half of a glucose by photosynthesis. 6, 3 more, 9 ATP per half a glucose, and a fair amount of reduced NADP. But that's okay, because where's all the energy that's used to make these things coming from? It's coming from the sun. And the sun is a pretty inexhaustible source of energy. So this Calvin cycle keeps cranking around, feeding carbon dioxide in, spitting out, if you like, half glucose molecules, and then that half glucose molecule, or G3P, can be used in other biosynthetic pathways, and we can make glucose out of it, we can make sucrose. The glucose can be used to make cellulose, the glucose can be used to make starch, the glucose can be used in anabolic processes to make fats. If you've got some nitrogen in the soil, the glucose can be used to make amino acids, so you can make proteins. It can be used to make nucleotides, so you can make nucleic acids. So the plant can really do it all if it has any input of sunlight, water, and CO2, and some minerals in the soil. Okay? Because this process is only producing carbohydrate, and other molecules require things like sulfur and nitrogen to start making amino acids. So those have got to be received from the soil. So plants, biochemically, can do a lot of things we can't do. Okay? So if you're out on the mall playing, frisbee or something like that, and you slide and get a big grass stain on your shirt, if you then happen to lay out in the ball for a while and catch some sun, will your shirt get sugary? Will photosynthesis happen? It won't. Okay? Why not? Well, you might say that, the chloroplasts are not part of cells. Okay? You just get that, mess of chlorophyll and probably broken chloroplast on your shirt, but it's not a cell. But in reality, you don't really need a cell to do photosynthesis. We used to do an experiment in this lab where we would take plant tissue and grind it up and then centrifuge it, do differential centrifugation and spin down the chloroplasts, and then take those isolated chloroplasts and put them into test tubes with a buffer and shine light on it, and those chloroplasts would photosynthesize. So really, chloroplasts, if they're provided the proper CO2 and water and sunlight, can photosynthesize even though they're not inside of the cell. Well, when you get that grass stain, you break open the cells, you break open the chloroplasts, and you don't have intact membranes anymore. If you don't have intact membranes, then you can't make ATP. If you can't make ATP, then you can't make this, even though your shirt is exposed to a lot of CO2. Okay? So this process happens in the chloroplast, and the chloroplast is remarkably adapted structurally to carrying out this process of photosynthesis the same way that the mitochondria was adapted to carrying out cellular respiration. So this might seem a little confusing, but in reality, there are just a few take-home messages that you need to have from this Calvin cycle, including its location, because that's relevant, the importance of this enzyme, and particularly in grabbing the CO2 and feeding it into the cycle, the understanding that this cycle requires energy inputs and hydrogen, that the output of the cycle has to equal the input, and that the cycle eventually has to regenerate this starting compound, this RUBP. Those are the big take-home messages of the Calvin cycle. About somewhere between 425 and 450 million years ago, plants made a transition from a strictly aquatic environment to the terrestrial environment. Longer ago than about 450 million years, plants couldn't survive on land. How come? Pardon me? Atmosphere was different. There was no oxygen to speak of, or at least not as much oxygen. No oxygen, no ozone. No ozone layer, ultraviolet radiation, gamma rays and stuff were bombarding the surface of the land. Water filtered that out. But as photosynthesis evolved and the oxygen atmosphere developed and the ozone layer developed, plants could then make that transition safely now from aquatic to terrestrial environment. So plants colonize land. Well, one problem of colonization had been solved by the evolution of the ozone layer. But there are some other significant problems that they face now. When you live in an entirely aquatic environment, internal support is not that big of a deal because water's pretty buoyant. Okay? So if you got these big, long algae, for example, they can float in the water. They can absorb a lot of sunlight because their blades are all erect. But what happens if you pull that out onto the land? It's all collapsed, right? So internal support was a big issue. What provides the primary internal support for plants, do you know? They don't have a skeleton like we do. Pardon me? They become turgid, that's part of it. But that only goes so much in terms of being able to make a tall plant. How about these big redwood trees, for example?
Just cellulose?
Cellulose is a part of it, most definitely. That supporting material around the cell wall.
Most of the tissue in the tree is alive.
Most of the tissue in the tree is not alive. It's actually dead tissue called xylem or wood. What does the xylem do for a plant? It transports water. Okay? And then there's another transport tissue for the sucrose that's moving around. That's called phloem. So that xylem and phloem make up an internal transport system or a vascular system, but that also provides support. So the evolution of xylem and phloem was a really important evolutionary step as plants moved from aquatic to terrestrial environments. There's another big issue plants faced once they went out of the water, besides support. Anybody know what it was? Predation was potentially a problem, but you could be preyed on in water too. Okay. Retaining water, yes. Dehydration. And as plants evolved, photosynthetic structures like leaves that were large and broad and flat, that was really great for having greater surface area for absorbing sunlight. But that greater surface area also was a disadvantage in that it was a greater surface area for water evaporation to occur from the leaf. Okay? So what was an evolutionary solution to water loss from leaves? The cuticle, that waxy layer that developed on the upper and lower surfaces. But what problem did that create? Photosynthesis. Now the cuticle prevented CO2 from getting into the leaf. So the solution to that then was these little stomata, these little holes in the bottom of the leaf particularly. And stomata are not just open holes. They can open and close. So if an organism were to live in a hot, dry environment, there potentially would be a great water loss from those leaves by evaporation, right? And that's not good for the plant. So, what would be a potential strategy under those conditions? Okay. Close those stomata, okay? If you close the stomata, then the water tends to be retained in the leaf. But, you close the stomata and what happens? CO2 can't get in, okay? So, if the stomata are closed, what's it like inside the leaf? What's our CO2 concentration going to be inside the leaf if the stomata are closed? It's going to be low because remember the Calvin cycle is using up any CO2 that got into that leaf. So CO2 would be a low concentration. What would the oxygen concentration be? Oxygen concentration would be high because do the light-dependent reactions require any CO2? No. So we would keep producing O2. Well, under these particular conditions, acronym here. This enzyme, rubisco, does something kind of destructive. It grabs O2 instead of CO2. And it sticks that O2 onto RUBP, this 5-carbon compound that begins the Calvin cycle. And when that happens, it breaks up that RUBP. And it leaves the Calvin cycle. If you start using this up, and pulling it out of the Calvin cycle, is that going to be good for carbon fixation? No. This process is called photorespiration. So plants are really in a bind because they don't want to get dehydrated. But if they close their stomata, their CO2 drops, their O2 increases, and this crazy enzyme that is just critical for the Calvin cycle to operate starts latching onto the oxygen instead of this RUBP, and we start oxidizing this stuff rather than converting it into sugar. And so the rate of photosynthesis goes down. Well, that would seem to argue that life in hot, dry environments, plant life, not very likely. But there are some evolutionary adaptations that occurred that allowed life to occur in these particular conditions. And these are sometimes referred to as alternative forms of photosynthesis. And the two that are discussed in the end of the chapter are C4 photosynthesis and CAM photosynthesis. These are basically evolutionary strategies to deal with this problem. Normal photosynthesis, if you like, this one right here, is called C3 photosynthesis. The reason it's called C3 is that the first stable product of this Calvin cycle is a 3-carbon compound. So most plants you're familiar with are C3 plants. All the trees on campus, all the little grass, shrubs and things like that have C3 photosynthesis. C4 photosynthesis includes plants like sugarcane and corn. Can photosynthesis are found in some plants that belong to a genus called Crasulaceae. These are plants that are called succulents. Aloe. I'll bring in my aloe plant on Monday. If you haven't seen an aloe plant, they have those thick, spongy leaves. Aloe is a succulent plant, and it has this CAM photosynthesis. So what the reading assignment does is it talks about how these differ and compare to the C3 process, and how these plants are a little bit different than C3 plants, sometimes structurally, okay? In one of these groups of plants, there's actually a little bit different organization of tissues within the leaves. There's a new type of cell called a bundle sheath cell, okay? So that's what the reading is going to discuss for your quiz on Monday. Okay? 5 minutes early. We're done with photosynthesis. Next topic we're going to be talking about is DNA. So when I graded this, initially what I would do is I would look at the diagrams, and that's why I circled this, and that's why I put this in there.
Yeah.
But then I read this and I went, okay. But so that's not why you missed points. There were a couple of reasons why I took a couple of points off.
Okay.
One of the reasons is that one of the really important things you needed to say was that this inner membrane is impermeable to hydrogen ions, because it asked about other properties of the
Okay.
membrane. Okay. You needed a little bit more detail here. I kind of saw what you were doing in terms of this, electron donor, electron acceptor things, and how that contributes to the hydrogen ion gradient. You didn't have FADH2 contributing to this, and then also the important role that the hydrolysis or the dissociation of water plays in the matrix. So there were a few little things. You had proton acceptors and donors, it's hydrogen acceptors. You didn't have your high hydrogen ions out here, you had your low ones here. Okay, so there were some little things here and there that took off a couple of points, but it wasn't because of the absence of that. I came back later and saw that.
Okay.
So I just wanted to explain that, but much greater improvement. That was
Oh,
great.
thank you.
I just wanted to remind you that I will be in class.
Okay, we will be doing something in class tomorrow that you can make up, and so just check with me when you get back on Monday, and I'll let you know what that is and how you can do it.
Okay.
Okay?
Okay.